Hi everyone, and welcome to our webinar today with Dr. Steve Chan, and he's going to be presenting to you the epigenetic regulation of breast cancer. My name is Rebecca Northeast, and I'm a product manager here at Proteintech, and I'm just going to do a really quick company introduction and also introduce Steve to you. So just a quick introduction about who Proteintech are as a company. So Proteintech is a leading manufacturer of antibodies, proteins, and immunoassays, and these are available across many different research areas. And our portfolio comprises of 3 core product lines. And through our Proteintech brand, we sell our antibodies and immunoassays. So we have antibodies against 13,000 human targets, as well as over 300 ELISA kits and IHC assay kits. Through our Humankind brand, we sell our cytokines and growth factors. And these are expressed in a mammalian cell line, in fact, a human cell line, so HEC293. And this means that they have native folding, post-translation modifications, and glycosylations. Through our Chromatech brand, we sell our nanobody-based reagents. And so these are excellent for immunoprecipitation, including for things such as CHIP, as well as IF and super resolution microscopy. Today, we're really excited to be working with Active Motif. And so Active Motif is a epigenetics company offering customers the solution for their entire chip workflow. If you have any questions or you want to get in touch with Active Motif, please do contact them on these emails below. Active Motif offers a full suite of products and services to assist scientists in their epigenetic research, thereby providing the tools to investigate the function, regulation, and interactions between genes. So today is all about the work done by Dr. Steve Chan. So Steve graduated with a BS in Sciences from Fudan University in China. He then did his PhD in a cancer biology lab at the in the McArdle Laboratory for Cancer Biology, and this was done at the University of Wisconsin-Madison in America. He did his PhD in Dr. Wei Xu's lab, where he is a postdoctoral research associate now, and I'm sure he's going to talk to you a bit about this later as well, but Steve is actually looking for a postdoc currently in another eminent chromatin biology lab. So if you know of one going, please do not hesitate to reach out to him, and I'll give you his details later. So that's it from me, and now to hand over to Steve, who's going to be talking to you about a recent paper of his that was published in a really high impact, in a really good journal. So thank you so much for agreeing to do this, Steve, and good luck with your talk.
Okay, thank you for the nice introduction, Rebecca. So it is my great honor to present my work on this well-recognized platform co-hosted by Proteintech and Active Motif. So our lab resides in the beautiful city of Madison, Wisconsin, where I spent my entire graduate school life here. So except for conducting scientific research, Madison is definitely an ideal place for vacation. Especially you are a fan of fishing, skiing, boating, and other outdoor activities. So today's talk is about my thesis work that applying some unbiased genomic and proteomic analysis to resolve epigenetic problems in breast cancer cells. You are also welcome to go through the details with the published article on the Nucleic Acid Research journal. So breast cancer is the leading cause of cancer death in women. And based on the gene expression profile, breast cancer have been classified into 4 distinct molecular subtypes, where ER-positive luminal tumors constitute about 70% of total diagnosis. And my study is mainly focusing on this subtype of breast cancer cell. The initiation and progression of breast cancer are controlled by a complex transcriptional networks in concert with extensive chromatin modifications. In particular, the ER-positive breast cancer, pro-proliferated pathways that drive cancer growth are activated by estrogen receptor alpha-dependent transcription. Estrogen-stimulated ER-alpha recruit many co-activators, which often carry histone modification activities to coordinate with basal transcription machinery to achieve productive cycles of transcription. Our lab has identified COM1 as a potent coactivator of ER-alpha, which specifically deposits the active histone marker X3 arginine 17 asymmetry dimethylation near the promoter of target genes. And this histone mark could further engage with RNA polymerase II associated factor complex, the PAF complex, to facilitate the transcription initiation and elongation for many ER-alpha target genes. So what do we know about the PAF complex so far? So the PAF complex is highly conserved and multifunctional complex regulating all stages of transcription, which was originally identified as an RNA polymerase II interacting complex in yeast. And CTLR9 here is the biggest key scaffold subunit in this complex, where depletion of CTLR9 will greatly impede the assembly and stability of this complex. Recent structural study show that CTLR9, shown in orange, is essential for the whole PAF complex integrity, where the alpha helix TPR domains of CTLR9 can serve as a bridge connecting the RNA polymerase II and other PAF complex subunits. And this multi-protein interacting model of the PAF complex agree with its potent coactivator function during the transcription by promoting the release and phosphorylation of the pause RNA polymerase II. The regulation of transcription elongation by PAF complex is not just simply interacting with the general RNA polymerase II proteins, but also controlling multiple transcription-coupled histone modification, such as H2B monoubiquitination, H3K4 and K36 methylation through interaction with respective histone modification enzymes. These unique coactivator functions of PAF complex, by coordinating both transcriptional machinery and histone modifications, give us great values to better delineate the transcriptional hierarchy that might mediate by the PAF complex in breast cancer environment. But why we want to study PAF complex or CTLR9 in only the ER-positive breast cancer? So we first carefully inspect the relative abundance of CTLR9 in different patient-derived tumor grafts with both ER-alpha negative and ER-alpha positive status. And these results indicate that CTLR9 is highly enriched in ER-alpha positive breast cancer. A more detailed mechanistic study carried out by our former PhD students further illustrate the important function of CTLR9 in ER-alpha positive breast cancer. Knocking down CTLR9 in those ER-alpha positive breast cancer cells erase over 90% of estrogen-regulated transcriptional response, demonstrating the function of CTLR9 in promoting the breast cancer, the ER-positive breast cancer progression. Given that the PAF complex or CTLR9 is important to incorporate many transcriptional active motifs, many transcriptional active histone marks during the transcription, as I mentioned before, such as the H3K4 and H3K36 methylation. So what happens to those histone modifications if CTLR9 was lost? Our preliminary studies support that CTLR9 knockdown will cause a noticeable decrease on those active histone marks, such as the H2B monoubiquitination, H3K4 and K36 trimethylation. But is it true that simply decrease of those active histone marks accounts for all the activation failure on over 90% of estrogen receptor responsive genes? Or there might be other histone modification changes contribute to this process. So we sought to achieve a more comprehensive understanding on how the global histone modification landscape is altered. To address this question, we employ a doxycycline-inducible CTLR9 knockdown cell line, where CTLR9 knockdown could be monitored by the expression of the EGFP reporter shown in the panel A. And Western blot results confirmed that treatment with doxycycline for 7 days was sufficient to deplete CTLR9. Beginning on day 8, doxycycline was removed for 0 day and up to 7 days to allow CTLR9 to recover to a similar level. To measure the relative amounts of histone modification, and total histone were extracted from the nuclear pellets of these CTLR9 inducible knockdown cells after treatment of doxycycline for 0 day and 7 days, followed by a quantitative mass spectrometry analysis. So as shown in panel B, the transcription coupled histone modification, such as H3K4 trimethylation, H3K36 trimethylation, would decrease by CTLR9 depletion as reported previously. But surprisingly, H3K27 trimethylation, a transcriptional repressive histone marker, was robustly increased. And these mass spectrometry results have been further validated by many other methods, such as Western blot ELISA. So it seems that the CTLR9 level is a key determinant for the cellular level of these H3K27 trimethylation. However, what happens if we re-expressing CTLR9 by removing doxycycline? Would the elevation of total H3K27 trimethylation levels caused by CTLR9 knockdown could be reversed? So to address this question, we perform a time course dox addition as well as dox removal experiment. So panel A shows that the H3K27 trimethylation levels increase when CTLR9 was depleted, and then return to its original level when CTLR9 was restored. The dynamic changes of H3K27 trimethylation in response to CTLR9 levels was further confirmed by the immunofluorescence staining shown in panel B. The intensity of H3K27 trimethylation staining in each individual cells was inversely correlated with CTLR9 levels, indicated by the EGFP reporter in a dynamic manner. So together, these data strongly suggest that CTLR9 is a regulator of cellular H3K27 trimethylation levels. So we know H3K27 trimethylation marks would deposit in a spatially defined chromatin domains to repress gene expression. But how CTLR9 refines the enrichment and distribution of H3K27 trimethylation on chromatin is still unknown. So we need to apply a quantitative ChIP-seq to map those increased H3K27 trimethylation marks caused by CTLR9 knockdown chromatin. So by analyzing the ChIP-seq data, we will perform some peak calling to obtain H3K27 trimethylation peaks. And those peaks we subject to some downstream analysis, such as the determination of peak location, as well as the peak distribution. Also, by performing the peak intersection among different groups, we can identify unique and overlap H3K27 trimethylation peaks. And lastly, we will compare the peak width and intensity, considering H3K27 trimethylation always propagate along the genome and forming a wide, broad peaks. Traditional ChIP-seq methodology, however, are not inherently quantitative, and therefore do not allow really correct comparison between samples with subtle difference. And moreover, variables such as different antibody specificity, the uneven immunoprecipitation efficiency, variation in genome fragmentation could further complicate the interpretation of the ChIP-seq results and make it really difficult to reflect the authentic biological relevance. To minimize the drawbacks of traditional ChIP-seq, a spiking strategy was developed in which equivalent amounts of exogenous spiking chromatin and spiking antibody are used to normalize technical variation during the sample preparation steps. So in this case, chromatin from Drosophila S2 cells is added, or you can call spike in, to each reaction as a minor fraction of the total chromatin. An antibody that recognizes the Drosophila specific histone variant H3K27me3 is also spiked into the reaction. So because of the specificity of the spiking antibody for the spiking chromatin modification, there is no cross-reactivity with the mammalian samples, so the background signal is low. So this is an example showing how traditional ChIP-seq normalization method might obscure the epigenetic difference. So in assumption, a cell population containing chromatin state A with high level of histone modification will appear similar to a cell population containing chromatin state B, where 50% of the signal has been removed, because the signal is quantified as a simple percentage of all mapped reads. But if you normalize the ChIP-seq signal by measuring the fixed amount of the reference epigenome, the epigenetic difference could be revealed to reflect its real biological relevance. So by applying our optimized ChIP-Rx protocol, we successfully determined the differential H3K27me3 methylation profile on chromatin when CTLA-9 was knocked down. So panel A depicts a cluster heat maps as well as a line plots of the average ChIP-Rx signals around the H3K27me3 methylation peak center with 5 kb expansion. The H3K27me3 methylation peak intensity in cluster 1, which represent those pre-existing peaks before CTLA knockdown, are similar between DOG 0-day and DOG 7-day groups. In contrast, upon CTLA knockdown, the intensity of the H3K27me3 methylation peaks would dramatically increase in cluster 2 as well as in cluster 3, which stands for overlap peaks with increased intensity as well as newly gained peaks respectively. Representative snapshot of the genome browser for each cluster shown in panel B also agree with the trend reflecting on this heat map that there are more newly gained peaks highlighted by those red triangles were detected when CTLA-9 was knocked down, as well as increased intensity on those overlap peaks highlighted by those black triangles. However, H3K27me3 methylation peaks are in general wide. It's making more sense to analyze specific genes putatively regulated by H3K27me3 methylation. So starting from the H3K27me3 methylation ChIP-Rx signal, we will perform kick calling to obtain both narrow and broad H3K27me3 methylation peaks. And each individual peak will be assigned to a specific gene through peak annotation. Where peaks reside in intergenic regions will not be considered. So we next annotate genes marked by H3K27me3 methylation in response to CTLA loss, where we found over 11,000 genes display at least twofold increase in normalized H3K27me3 methylation ChIP-Rx signals after CTLA was knocked down, while the signal was drawback upon CTLA was restored. The H3K27me3 methylation is solely established by this PRC2-EZH2 complex in human. And given that loss of CTLA-9 results in a significant increase of H3K27me3 methylation peaks intensity on chromatin, so we examine if CTLA loss also affects the PRC2 chromatin recruitment, especially on those putative H3K27 trimethylation regulated genes. So we again performed the ChIP-Rx using an antibody against SUZ12, the quarrel subunit of PRC2 in CTR9 inducible knockdown cells under the same treatment condition. To determine how CTR9 refines the global PRC2 distribution on chromatin, we allocate the SUZ12 ChIP-Rx signal on those putative H3K27 trimethylation regulated genes shown in panel A. And over 11,000 putative H3K27 trimethylation regulated genes concurrently display a dynamic SUZ12 changes in response to CTR9 levels by DOX treatment, as depicted by an example captured from the genome browser shown in panel B. So this data collectively suggests that increased PRC2 binding, at least in part, accounts for increased H3K27 trimethylation deposition in response to CTR9 knockdown. To investigate if genes harboring CTR9 binding sites are more likely to be subject to H3K27 trimethylation regulation than genes without CTR9 binding. So we sought to map CTR9 genomic binding sites and derive CTR9 regulated genes from ChIP-Rx binding peaks. Because the chip-grade CTR9 antibody is not available, so we first generated a clonal breast cancer cell line with a 3 times FLAG epitope tag knock-in at the start codon of CTR9 using CRISPR-Cas9. The Western blot result here showing that the FLAG tag CTR9 protein was successfully incorporated into the PAF complex. To identify putative genes regulated by CTR9, we perform a ChIP-Rx using commercial FLAG antibodies from 2 different vendors. 6,750 genes were identified by overlaying the FLAG CTR9 binding peaks associated genes derived from 2 different FLAG ChIP-Rx data. Over 70% of genes containing the FLAG CTR9 binding peaks overlap between the 2 antibodies. And only the shared peak genes were considered as a bona fide CTR9 binding genes, target genes. Interestingly, PRC2 proteins such as SUZ12, EZH2 were predicted as the cis regulatory factors enriched on those putative CTR9 regulated genes. And around 50% of those genes with FLAG CTR9 binding peaks overlap with those harboring increased H3K27 trimethylation peak intensity. And the results here show that the CTR9 binding genes are highly responsive to the H3K27 trimethylation and PRC2 regulation. So in a short summary, CTR9 knockdown leads to increased total H3K27 trimethylation levels, as well as the genome-wide expansion of H3K27 trimethylation peaks, depicted by increased intensity as well as increased peak number. Also, CTR9 knockdown enhanced the PRC2 chromatin association. And lastly, CTR9 target genes are likely regulated by PRC2 H3K27 trimethylation signals. So we next want to ask if the enhanced PRC2 recruitment on chromatin is the only reason account for the dramatic genome-wide increase of H3K27 trimethylation when CTR9 was knocked down. Is it possible that CTR9 also affect the enzymatic activity of PRC2? So many studies support the existence of 2 PRC2 complex subtypes in mammalian. The subtype is determined by the auxiliary proteins associated with the coral PRC2 complex. EPOP or PALI and one of the polycomb-like proteins are found in the PRC2.1 subtype, while AEBB2 and GERI2 are found in PRC2.2. And those auxiliary proteins shows differential capability to either promote the PRC2 catalytic activity or facilitate its stability on the chromatin. So therefore, it is necessary to examine if CTR9 affects the subunit composition as well as the chromatin association between these two different PRC2 subtypes. Emerging evidence illustrates that two mutually exclusive PRC2 subtypes could antagonize each other and elicit different capability for H3K27 trimethylation deposition and propagation of these marks to form or repressive domains, especially in many stem cell contexts. But all these evidence suggests that the PRC2.1 subtype might deposit H3K27 trimethylation marks and expand those repressive domains in a more potent manner than the PRC2.2. Then we begin to wonder if loss of CTR9 will shift the PRC2 balance from PRC2.2 to a more active PRC2.1. To validate the loss of CTR9 is sufficient to alter the PRC2 subtype balance. So we perform a glycerol gradient sedimentation to separate two PRC2 subcomplexes from the nuclear fractions based on their difference in molecular weights, where the calculated PRC2.2 molecular weight is nearly 100 kilodaltons larger than the PRC2.1. So when CTR9 was knocked down, the PRC2 complex changed from the heavy form to the light form of PRC2.1. In addition to the protein abundance change, we also perform a ChIP-Rx for GERI2, a PRC2.2 specific subunit, and MTF2, a PRC2.1 specific subunit, to address if the PRC2 subtype switch is also occurred on chromatin. So upon CTR9 depletion, decrease GERI2 and increase MTF2 genomic association was observed across the SUZ12 peaks, shown in both panel A and panel B. And these data collectively support the model that CTR9 loss triggers the expansion change of the PRC2 facultative subunits, leading to a switch from the enzymatic less active PRC2.2 to a more active PRC2.1, which facilitate the propagation of H3K27 trimethylation repressive domains. In another short conclusion, CTR9 knockdown leads to a switch from PRC2.2 form to PRC2.1 subtype. Loss of CTR9 decreased PRC2.2, but increased PRC2.1 chromatin association. And the last point, which I didn't show you today, but you are welcome to look through my published paper, that artificially manipulating the expression of PRC2 subtype by overexpressing PRC2.1 or knockdown PRC2.2 could partially recapitulate the genome-wide increase of H3K27 trimethylation caused by CTR9 knockdown. Mutations in subunits of PRC2 have been increasingly identified in multiple cancers, which confer cancer cells with different dependency on H3K27 trimethylation and PRC2, which indicating that pharmacological inhibition of PRC2 could be a valuable therapeutic approach for treating cancer. So EZH2 inhibitor has been widely used to suppress the triple-negative breast cancer tumor growth and metastasis in many preclinical models due to the increasing TMBC cell plasticity of differentiating to a more luminal-like cell type. Our data that the TMBC cell lines display higher levels of EZH2 and H3K27 trimethylation levels support the application of PRC2 inhibitors in treating the TMBC cells. So given that the EZH2 activity as well as the H3K27 trimethylation levels was significantly soar when CTR9 was lost in ER-positive breast cancer cell. So we raise up an interesting question. Could EZH2 inhibitors also be applicable to CTLA-9 low expressing but ER-positive luminal breast cancer? So to address this question, we first figure out if CTLA-9 affects the breast cancer cell sensitivity against the EZH2 inhibitors. And in our study, we apply a potent EZH2 inhibitor, UNC1999, which is already bioavailable, and UNC2400, a structurally similar but inactive analog compound, to exclude the possibility of off-target effects. The cell viability was first measured by the MTT assay. So as expected, CTLA-9 knockdown cells were more sensitive to UNC1999 than the parental cells. And the EZH2 knockdown cells were included as the negative control. Moreover, this hypersensitivity phenotype is not only restricted on the 2D molar layer culture, but also in 3D tumor spheroids model. So in this experiment, we employ a dual fluorescence-based cell cytotoxicity kit to quantify the differential cytotoxic response between control knockdown and CTLA-9 knockdown 3D spheroids towards these EZH2 inhibitors. So when treated with ascending concentration of these UNC1999, the CTLA-9 knockdown 3D spheroids elicit a stronger cytotoxic response than the control group, depicted by more red dead cells and less green live cells. And UNC2400 serves as a negative control. In addition, treatment with other EZH2 inhibitors in different ER-positive breast cancer cell line also shows similar results. So in summary, CTLA-9 depletion rendered the cells more sensitive to the EZH2 inhibitors, as measured by the inhibition of growth, proliferation, as well as induction of cell death in ER-positive breast cancer cells. So this uncovers that the H3K27 trimethylation levels are precisely controlled by CTLA levels in breast cancer cell. A global quantitative analysis of histone modification revealed a significant increase of H3K27 trimethylation total levels, as well as the genome-wide spreading of H3K27 trimethylation peaks upon depletion of CTLA-9. This is partially attributed to the increase of PRC2 recruitment, but more significantly, to switch from PRC2.2 to PRC2.1, of which PRC2.1 has stronger H3K27 methylation activity and propagation capability. Genes harboring the CTLA-9 binding sites are often found to process the gain of H3K27 trimethylation repressive signals upon loss of CTLA-9. And finally, CTLA-9 depleted cells become addicted to H3K27 trimethylation and are hypersensitive to PRC2 inhibition. So collectively, our study uncovers a unique mechanism by which a transcriptional elongation factor demarcates the PRC2-mediated H3K27 trimethylation domains by regulating the PRC2 subtype balance in breast cancer cells, and provides a molecular basis for therapeutically targeting CTLA low breast tumor with EZH2 inhibitors. So for take-home message, our findings of CTLA-9 demarcates the PRC2-mediated H3K27 trimethylation levels and genomic distribution provide a unique insight as to how transcriptionally active states are converted to repressive chromatin regions. So in the presence of CTLA-9, the predominant form of PRC2 is PRC2.2 to maintain a basal level of H3K27 trimethylation level. Depletion of CTLA-9 causes a general transcription blockade, followed by an increase of H3K27 trimethylation levels and expansion of these repressive domains, which are likely results of enhanced recruitment of PRC2 to chromatin, as well as the PRC2.2 to PRC2.1 switching. So at last, I need to express my gratitude towards different groups of people, and I need to thank my advisor, Professor Wei Xu, for all her patience, guidance, and support. And secondly, I want to thank my lab members for creating a really fantastic environment for scientific research. And I would especially like to recognize the time and help I received from our PhD candidate, Christine, who spent a great deal of time proofreading and editing my manuscript and my presentation slides. And my appreciation also goes out to Dr. Gui Ma and our former lab member, Dr. Hausen, who helped me construct and validate many important cell lines and reagents. And I would also like to extend my thanks to our collaborators, were Dr. Junfeng Huang and Professor Lingjun Li for performing the mass spectrometry analysis, and Dr. Sitan for sharing Drosophila S2 cells and help me optimize the ChipRx experiments. And again, thank you so much for all of you attending this webinar, as well as the organizer from Proteintech and ActiveMotif. If you are interested in my work, you can send me an email for more detailed discussion. Also, our lab is now looking for ambitious and motivated postdoc. If you're interested in our lab, please feel free to contact Dr. Wei Xu. And also, as Rebecca mentioned, I'm also actively seeking a postdoc position. If any lab are looking for a postdoc and working on epigenetic, chromatin, transcriptions, please feel free to let me know. And I'm happy to take any question you have. Thank you.
Hi, Steve. Thank you so much for such an interesting talk. You went through every aspect of your talk in a really detailed manner, and I, as a non-epigenetic scientist myself, I actually felt like I learned a lot. Thank you so much. If anyone's interested in asking Steve any questions about his research, please do type your questions either in the chat box or in the Q&A section. And we do have a question from Yicheng Long, who says, thank you for the nice talk. Besides MTF2, I'm wondering if you have checked the expression level in chip signal for other PCLs, such as PHF1 and PHF19.
Yes, we did. We found that MTF2 has the highest expression than other PCL proteins. PHF1 can hardly be detected in our breast cancer cell. But PHF19, we can also detect a moderate levels of PHF19. But MTF2 associated PRC2.1 is the dominant form in breast cancer cell.
Thank you. That's a really good answer. And another question here is from Rashmi, and she asks, hi, Steve, lovely talk. Have you looked at patient-derived organoids?
Well, that's a really interesting aspect. So if ideally we want to test that CTLA-9 might be serving as a biomarker to predict the EZH2 inhibitor sensitivity, I mean, applying patient-derived organoids will better answer the question. But unfortunately, so far we haven't applied those patient-derived organoids. But in the future, we definitely want to look at this relationship between CTLA-9, PRC2, as well as H3K27me3 trimethylation in many patient-derived organoids or any patient-derived xenografts.
That's great, thank you. We have a lot more questions coming in here. So another one from Takeshi, and he asks, how are CTLA-9 protein levels and activities regulated in ER-positive breast cancer cells? And in addition, have you checked the expression of CTLA-9 in other cells?
For your first question, that how CTLA-9 was regulated in ER-positive breast cancer cell, my straightforward answer is I don't know, but my best guess is they're also subject to many epigenetic regulation. So we need to do a more histone modification and other chromatin remodelers profiling by using different genomic sequencing to see whether CTLA-9 genes itself harboring different histone modification or other chromatin remodelers, or even we can map some specific transcription factors binding site. So no one actually so far has reporting how path complex or how CTLA-9 genes was regulated. So for a second question, have you checked the expression of CTLA-9 in the other cells? I think in one of my slides that showing that CTLA-9 is highly enriched in many ER-positive breast cancer cells, including MCF7 cells, T47D cells, BT474 cells. And also, I repeat all these phenotypic measurements on H3K27me3 as well as the PRC2 subtype in different ER-positive breast cancer cells, and they all show similar results.
Okay, thank you for your answer. And then we have a relatively long question here from Sebastian. And he's asking, PRC2 is known to be able to bind to RNA and be stolen from the chromatin. Therefore, how do you know that the transcription blockage is sufficient to increase PRC2 occupancy at the chromatin? Have you tried to perturb transcription, e.g. using RNA polymerase II inhibitors, and do you see if this is sufficient to induce the PRC2 subtype switch?
So for the first question, so PRC2, yes, have a really high affinity for RNA binding, especially for those RNA with intact poly A tail. So in our scenario, when you knock down CTLA-9, it affects the whole path complex integrity. So the whole path complex could not function properly along with the RNA polymerase II during transcription. And we have a lot of evidence suggests that knocking down CTLA-9 perturbed the transcription initiation, the RNA polymerase pause release, as well as the transcription elongation. So that's why there's no more abundant amount of poly intact messenger RNA to inhibit PRC2 occupancy on chromatin. So that's why PRC2 are more likely to sense those perturbed genes with impeded transcriptional activity, and then deposit H3K27me3 methylation marks to safeguard the chromatin integrity. And the second question is, do you try to perturb transcription and see if this is sufficient to PRC2 subtype switch? And yes, we did do some preliminary data, and by treating the cells with DRB, which is a potent elongation inhibitor, and we indeed found that there is a loss of CTLA-9 binding after treating with DRB, and also an increase of H3K27me3 methylation and PRC2 binding on a large fraction of those we previously mapped H3K27me3 methylation putative regulated genes. But we haven't yet to test whether DRB treatment also caused a sufficient PRC2 subtype switch, but that's something definitely we want to test in the next step.
Great, thank you. And finally, we have one more question, which is breaking down into two about the techniques you use. And Martina as well, if Steve hasn't got the necessarily all the answers for you, then please do feel free to reach out to someone at Active Motif. I'll give you their email and they'll be also to help you as well, because I feel like this is just a generic technical question that the specialists at Active Motif will be able to help you with. Yeah. So she asks, can ChipRx be used for a low amount of cells?
Unfortunately, it's no. I would say because ChipRx is optimized based on the traditional ChIP-seq protocol, which require a larger amount of cells. I would say about 30 to 50 million cells to ensure you have enough high-quality reads on the sequencing instruments. But if you think if you want to start with low cell numbers or some cells that is hard to harvest, I would suggest to switch to cut and run or cut and tag technologies, which only require less than 50,000 cells to generating a really great genome-wide histone modification or other transcription binding profile. So that's my answer.
Thank you. That's really great. And like I said, please do feel free to contact any of the product managers or sales team at Active Motif, and they'll be able to help. Martina also asks, and it sort of goes back to your answer, Steve, have you tried to use cut and run in these experiments?
No, unfortunately, but we are now actively optimizing the cut and run protocol with another lab in our department, and to see whether the cut and run can be sufficient to apply different cancer cell types or different patient-derived primary results.
Okay, great. And thank you so much, everyone, for submitting your questions. And I think what I'll do now is just finish with some more company slides. But I just want to say again, thank you so much to Steve for such a good talk. I really enjoyed it, and I think you went through the data in an incredibly concise and clear manner. And I really would like to encourage everyone to reach out to his lab. So either reach out to Steve himself, you can see his email is down here, or please do, if you are interested in that postdoc, please do reach out to his boss as well. But again, thank you so much, Steve. And like I said, I'm just going to quickly share my screen and go through a couple more slides about us as a company, and then give you some contacts as well if you want to get to if you want to hear anything from us. I just want to let everyone know that as part of attending today, you will receive a personalized certificate of attendance, and also sorry, there we go. You'll receive a personalized certificate of attendance, and also any way that you will also provide you with a form so that you can try a free sample of our antibody or our nanobodies as well. So just quickly, Proteintech antibodies are all made in-house, and they undergo extensive QC by our R&D scientists in-house as well. And so that actually differs from many other antibody companies out there, whereby they get their product from many different manufacturers. We also have over 140,000 citations to date, and this is really credible to you and people such as Steve who publish with our research, and it's really great that we can see our products are being used in so many different people's hands. We have antibodies against 13,000 human targets, and this is the largest range of in-house manufactured antibodies in the industry. And over 3,000 of our antibodies are, of our targets, are validated by knockdown-knockout genetic validation. And as well as this, I feel like this audience is interested in a lot of epigenetic studies. 4,000 of our antibodies have been validated for IP, and 220 of these have been published for CHIP. And so we really truly believe that we can help you with your epigenetic research. As well as through our Proteintech brand, we also have our Chromatech brand. And through Chromatech, we are able to sell our nanobody-based reagents. And so a nanobody is simply the binding domain of a heavy chain camelid antibody, specifically an alpaca. And so what we can do with these nanobodies is we can conjugate them to beads, and they become what are called nanotraps. And these allow for really efficient pull down and detection of proteins in an IP format. So they allow fast, reliable, and efficient one-step immunoprecipitation. It means they have less background and no unspecific interactions, as there is no protein A or protein G involved. And also, due to their really high affinity, they have our GFP trap has an affinity of 1 picomolar, which is incredibly high for an antibody. This enables the precipitation of really low protein amounts. These nanotraps are also really useful in things such as CHIP. So we have many different application notes and publications of our nanotraps being used in CHIP. And we have these available for multiple tags, predominantly for GFP, mCherry, flag tag, MUC, and V5. So like I said, everyone who attended today is able to claim a free sample from us. We will send out a link for you to be able to download this when we do our follow-up. We'll also send a recording of this talk as well, in case you missed any parts, or you want to forward it on to your friends who potentially missed it. We'll also enable you to try a free nano trap. So again, one of our GFP traps, which is excellent for IP and CHIP. So just going to leave that there, and thank you so much for joining, everybody. I really can't thank Steve enough for this. He gave such an excellent talk, and he answered the questions really well. And if you have any questions regarding Proteintech, then please do email either myself or our events email. Or if you have any questions regarding the active motif, any sort of technical questions or any product questions, please do reach out to Mike Gabati, who, due to the fact that he's in California, wasn't able to join today because of the time difference, but he is more than happy to answer any questions that you might have. So again, thank you so much to everyone for listening. And like I said, we'll send you a follow-up email with all the details that you might need. Thanks again.